Today, topic of the presentation is compartmentation of metabolic pathways. Already I have discussed about the allostatic regulation and activation of latent enzymes in my last two videos. You can check those videos. Today in this video, I am going to discuss about compartmentation of metabolic pathways. This is also one of the important ways of regulation of enzyme activity in the living system, similar to allostatic regulation and activation of latent enzymes. Already I told you that enzymes are also regulated by compartmentation. It mostly occurs in eukaryotic cells. For eukaryotic cells to function properly, they divide their intracellular space in subcellular compartments, each harboring specific metabolic activities. Compartmentalization of metabolic pathways is a prerequisite for certain cellular functions. During compartmentalization, enzymes are separated by cellular membranes that restrict their movement to a certain compartment. Those are known as cell organelles. This means such enzymes are only able to function inside the compartment and will not be able to reach substrates outside the compartment. This method of regulation can also provide the right environment for enzyme activity to occur. Let's know why compartmentation is important. Compartmentation provides optimal conditions for enzyme activity. You know that enzymes catalyzing individual reactions often have different temperature and pH optimums. By dividing the cellular space, that means compartmentation, optimal conditions for individual enzymatically catalyzed reactions are created. As for example, enzymes localized to mitochondria can reach an extreme internal environment as compared to the rest of the cell. These conditions inside the mitochondria, that means a relatively higher temperature and pH, allow the functions of certain enzymes to be enhanced. Compartmentation is important in order to protect against the activity of lytic enzymes. For example, sealing the cellular digestion in lysosomes prevents an unwanted autodigestion of other organelles within the cell. A common processes that accompany the disruption of some of the compartments, like spilling the content of lysosomes or mitochondria, are necrosis or activation of apoptosis. Apoptosis means the process of programmed cell death. So in that case, compartmentation is protecting from necrosis and apoptosis. Compartmentation affects the regulation of metabolic pathway as well, making them more accurate and targeted and less interfering with each other. Generally, the anabolic and catabolic pathways are operated in different cellular organelles. That means in different compartments to achieve maximum economy. There is no point for simultaneous occurrence of both the pathways. As for example, enzymes for fatty acid synthesis are found in cytosol, whereas enzymes for fatty acid oxidation are present in the mitochondria. Different metabolic pathways which are occurred inside the cytoplasm are glycolysis, hexose monophosphate shunt, fatty acid synthesis, purine and pyrimidine catabolism. And inside cytoplasm, all the enzymes involved in these different metabolic pathways are present. In mitochondria, all the metabolic pathways are fatty acid oxidation, amino acid oxidation, Krebs cycle, urea synthesis, electron transport chain, and oxidative phosphorylation. So all the enzymes involved in these metabolic pathways, except in case of Krebs cycle, succinate dehydrogenase is located in the inner mitochondrial membranes in eukaryotes and in the cytosol in prokaryotes. Inside nucleus, biosynthesis of DNA and RNA is occurred. So in that case, all the enzymes involved in biosynthesis of DNA and RNA are present inside nucleus. Inside endoplasmic reticulum, cytochrome P450 esterase, these enzymes are present, and metabolic pathways are protein biosynthesis, triacylglycerol and phospholipid synthesis, steroid synthesis and reduction. These are occurred inside the endoplasmic reticulum. Inside lysosomes, different digestive enzymes are present like lysozyme, phosphatases, phospholipases, hydrolases, proteases, lipases, and nucleases. Inside lysosomes, hydrolysis of proteins, saccharides, lipids, and nucleic acids occurs. Inside Golgi apparatus, glucose 6-phosphatase, 5C nucleotidase, glucosyl, and galactosyl transferases, etc. enzymes are present. And metabolic pathways are post-translational modifications, protein shorting, and formation of secretory vesicles. Inside peroxisomes, catalase, uretase, urate oxidase, D-amino acid oxidase enzymes are present, and metabolic pathways are long chain fatty. Metabolic pathway is long chain fatty acid oxidation, which is occurred inside the peroxisomes. Although compartmentation is very important, at the same time it puts greater demand on the energy consumption. It arises from a frequent need to use ATP-dependent transporters, transporting substances across membranes against the concentration gradient, and thus creating different environments in different compartments. Thank you very much for watching this video. In order to motivate me, kindly like, share and subscribe this channel. Don't forget to press the bell icon to get notifications.